the speakers with the wonderful talks, and I think everyone deserves a prize. And also should thank the University Scholars Funds because they provide for your iPad minis. So what it means is that you must use them only for scholastic activities, nothing else. So I gave a title, but I changed my title because many of the people here are new to the groups, just joined the lab in the last couple of years and so on. So I just wanted to tell you about the history of the two laboratories from my own perspective, mainly for fun. So that's Hammy retreat here. The question first was, how far do I want to go back? I could go back to the period when I can remember myself and so on, but I thought I'd just go back to my college years because that's the period you can still remember, right? So that is me as a freshman, and so that winter five of us went to British Korea for 2 months to study physics, nothing else, just study physics. And then 2 of them left in 2 weeks. So we stayed for the entire period. And so one day we were really bored, so we just hung out and then took a picture.
So this is DJ.
And then DJ comes to
Is it a manual?
Is that MR2, is it?
I can get it.
So I came to Berkeley as a grad student in physics, and then I bought a used sports car.
It's a MR2, is it?
Yes, MR2. And it has an engine in the back. So it's a 2-seater. The engine is so loud.
I have it. You.
And so then I met Suwa, we dated, and she hated the car because of it's stick shift, and then for me it was a lot of fun. And then I joined the Shemla lab as a grad student. Jan Shemla's father, Daniel Shemla. So that's me. Huh? Yes.
You have the same t-shirt, different color.
Then, so we got married when Suwa was still in college, when she was a senior in college, and then this is in Hawaii. So I rented a Mustang. First child, Gloria, was born in 1996. Then second child was my first paper. So finally on my PhD, I finally published one paper. So that was the demonstration that you can measure thread between a single donor and single acceptor molecule. Published in PNAS after a couple of rejections from other journals. And you can see Paul Selby here and Shimon Weiss there. Shimon Weiss was a staff scientist in the Shemla lab, but he was my effective advisor. I talked to him every day. I think I talked to Daniel Shemla maybe 5 times during my PhD. So that was the thread paper, but you can actually see that it mentions that near field scanning optical microscopy was used to obtain two-color image and emission spectra from donor and acceptor. So it's useful to talk about what ENSOM was. That was the super resolution imaging method 20 years ago. Very popular. Many people were doing this. In fact, many people who were in that ENSOM community then moved to single molecule fluorescence and then really pushed the field forward. So we are the latecomers in that community. So this is how it happened. So I was a grad student at Berkeley, and in early 1993, Shimon Weiss told me that read Eric Batchik's. Eric Batchik is a famous biophysicist that invented PALM method, this the other super imaging method. But he was actually pioneering the ENSOM method at the labs. So he told me to read his PhD thesis and then study, build ENSOM and study semiconductors. So I did. But it wasn't useful for semiconductors at room temperature because of many reasons. So I was stuck, right? And then Shimon said, well, look at what Batchik and Sanyizian and Peter Ambers published. They just published that you can show see single molecules at room temperature using ENSOM. So it's cool and it's hard to rescue single molecules. So okay, but just doing single molecules isn't new anymore. How then can I get a PhD? So that was my reservation. And then he said, oh, let's do two molecules. That's how we did Fred. But then, wait, but how do we put two dyes close to each other? Well, their concentration is low enough, you can see single molecules. So we asked a lot of people. In fact, initially I just spread two molecules randomly and tried to find some randomly colocalized molecules, but it was pretty hopeless. And then someone told us that there's a guy named Paul Selvin on campus who can use DNA to link the two dyes, bringing them close to each other. So we talked to him. He said, oh, we can help you. I can help you, but also this is useful for biophysics. So that's how we began to do collaboration and so on. You can see that Paul Selvin is here, Yan Chemla is here. So, all these connections when you are very young can become very important, really shape your career and in your life even, okay? And then I went to Stanford to join Stephen Chu's lab right after he got the Nobel Prize. That was very nice because he was very busy, so he didn't bother me very much. And in this picture, taken when Steve Chu became 50 years old, his birthday party, I'm here. That's Xiaowei Zhuang. And who else do you know? Harold Kim is now Professor at Georgia Tech. Doug Smith is Professor at UC San Diego. Hazen Dabcock is married to Xiaowei Zhuang. And Vladimir Gledic is a professor at MIT. He's a professor at the University of Chicago, and so on. Okay, so it's a pretty good group of people. And then I got a job in Urbana, so started a new lab. So this space now is Ender 1, Ender 2, and this, my home lab. Used to be a machine shop, so they vacated the machine shop to make room for my lab. The machinist, before he left, he cursed me. He said, you use this as a machine shop for 20 years, 30 years, whatever, so all the machining oils, seep into the floor, so it's going to continue to come out, right? And then, so I was worried, but then when they open up the floor, there's nothing there, so. So that's, now this is an area that is now occupied by Chu Young and some of you guys as a cubicle space. So that became my first lab space because the other side of the door was under remodeling. So we just put a laser table, side building, a microscope there, and people separates the wet lab space from the laser lab. So actually it was built, one by one this way. Interesting. And then first group members, Greg Okumus, Ivan Raznik, and I forgot his name. Yeah, he left my lab after 6 months, so he's forgotten. Yeah. James? James Adrian.
No.
He also left.
Yeah, he told me after 6 months that he doesn't like to use pipettes. He hates anything wet, so he went to theory. Yeah. Then the lab is almost ready. You see now the wall is complete, and Ivan is now doing experiments in that temporary space. The lab is now really ready. Can you recognize the space?
Jelly.
There should be 75 gels stacked
better
in the ceiling.
Jelly.
And new students joined, Kathy and Sean. They've joined as a zero-year summer students in my first summer here. And then somehow they were attracted to each other, and then they got married to each other later. And I was the last one to know in the lab. And Sua joins. That's Ben Stevens. And Gloria joins. And it's a full house. So that was actually when Sua was expecting our second child, Stephen. So it was a baby shower in the lab. There's the first group retreat. Ellerton Park. It's not really bright enough here, but so Greg Okumus saying that you can do better than circadian immobilization by doing basic encapsulation, and so this is talk, and these are invited guests from the Saving Lab who came to our retreat.
Is that on?
Yeah. And this is lunch at the retreat, and Gloria and my mother who came too. It's a family business.
Gloria gave a talk.
Yeah, Gloria gave a talk too. Yeah. This was 3 scientists. Yeah. And it was a beautiful day outside. And Sua labeled the replicates successfully. So she made a clay model to demonstrate the conformational changes that she was measuring. And Stephen came. So actually one day, Sua told him that with the Pikachu pillow, you look just like a Pikachu. And Stephen said that, oh, I'm not Pikachu, I'm Stephen Chu. So actually you can watch a YouTube video where I show this slide in Steve Chu's birthday party. And then HHMI happens, so we had a big party in my old house, and actually my group members then did grilling, so yeah, I was cheap, yeah. And then inside, while these guys are hard at work, so I was singing. This is our third group retreat in Wisconsin Dale. I'm pretty fair skin. That's Ibrahim and Rahul. of the activities back then was to go to a pottery store, and then basically we painted our own potteries. That's Charlie and Babu. And then technician is a foreign concept in a physics department. We don't have a technician in physics, but because I got HHMI, I hired a technician. So Salman joined the lab that year. And this was a bachelor's party for Ivan, who was getting married. So we had to play golf. That's Reza with first of the many girls he wrote. Then following was complete, so that was a picture of that day. One year I went to Brazil for a biophysics meeting, and then somehow members in Ghana applied to the meeting, and they got actually accepted to the meeting, and then they paid for everything for all of them. I didn't even have to pay anything for. So we went to Brazil together, and Ibrahim. Looks like maybe it's elongated. Okay, and JJ. And then this is Candice Tanner, who was a student in the Graton lab here in the physics department, and she then went on to do postdoc with Mina Bissell at Berkeley, and she is now PI at NCI, National Cancer Institute at NIH. And she just hired a student from her Sabin lab, Ben Lang, that you know, is joining her lab at NIH. And I met this guy in Brazil, and he was very smart, so I really liked him. So I asked him to join my lab as a visiting student. But it turned out he was an undergrad. So he came to my lab for 2 months. You remember him? Victor? This guy?
Yes.
Yeah. He worked with Eugene, yeah. And then he went to grad school in the Netherlands with Antoine Van Ooyen. So, it's excellent meeting. This kind of meeting can also lead to something actually substantial. And then number 3 arrives, my third child. So you can see that he arrived January. Actually, this is not his birthday. This is one day after his birthday, January 08. So, so in the same week, we published a paper in Science Express on Vigai translocation. So when Peter was born, we made this basically a New Year's greetings card saying that due to 2 major collaborative projects we have been working on. So one was a paper, the other is a baby. And then someone told us that, he likes one of them, but not the other one. That was not Phyllis. And then Myong lab starts. Okay, it's that year. Why is it sometime?
Yes.
Okay. So I stayed until 1:30 last night to find all these photos. I think it's really interesting that the photos become bigger and bigger inside, file size, as time progresses. Right. So let me show you some. That was Gloria in 2003 demonstrating the hand open mechanism for myosin 5 moving on actin filament. in the myosin 5 experiment, what you are doing is to put a dye on the protein, and then image the protein moving down the track in the middle of the light. So that this is a famous skier, Lindsey Vonn. But if you just have one dye only, then you cannot really learn how come she's so good, right? So what you want to do is reflect, right? And so that's Steven.
Yeah.
Like him from the moon, you cannot see the two lights as two separate lights. What you see is a single light with different color, depending on the conformations. Right? So that's the idea. So I want to show you some real data. So DNA is damaged very frequently in your life, and can result in many different problems. And 1 light year is the length of DNA that you make in your lifetime. 1 light year is not the unit of time; it's a unit of length. How far the light travels in 1 year. So that is about 10 to the 14, or 10 to the 16 meters. And then in your body you have about 10 to the 14 cells. So if you account for the fact that you have 2 meters of DNA per cell, then you're also making many cells again and again every day. You make 10 to 11 cells, blood cells per day, 10 to 11 immune cells per day. So that approximately corresponds to 1 light year. So to keep the DNA intact, when you make so much DNA, you have to have DNA repair system. And one way to do that is to use recombination. And recombination is about as close as you can get to having sex, if DNA can have sex. Okay? So damaged DNA has a blunt end cut, and then enzymes can digest the DNA into a single strand that can then search for homologous DNA in the cell to start the recombination. But this process is inhibited by protein filament, by red K protein. And the question is that, how do you find this matching sequence when you have millions of different base pairs in the cell? How do you find that matching sequence very quickly? So if you want, this is equivalent to finding a soulmate. For those who are single, how do you find your perfect partner in a short time? Okay. So one idea is this. Basically, you try to find the soulmate through a random 3D search. Basically, you just date a random person on the street. Okay. So then it's not a match. I come up, and it's not a match. I come up, right? And then you do it. Okay. And you did many millions of times until you find the right match. But that can be very inefficient. Okay. Actually, it worked this time, but for much longer DNA, that's very inefficient. So the other possibility is called 1D sliding. So if you want, you can think of it as, okay, instead of dating a random person on the street, let's focus on small group of people. So let's join a knitting club. Okay? Then you have 20 people in the knitting club who have the same hobby. Then you can sample that club. So that is the 1D sliding. You find and then slide, and then you find the target. So it turns out that this is how it works according to our data. So as Kaushik's paper in E-Light last year, so you put a dye on the protein filament, form a single-strand DNA, and you put another dye on the double-strand DNA, and there's no matching sequence. There's no matching sequence. So it's like, then what happens is that you see flat fluctuations because it can bind and then slide, the filament can slide on the double-strand DNA, trying to find the homology but not finding it. Okay? The question is that, what can this sliding lead to actual identification of homology recognition? So what we did was to put two matching sequences at two different sites on the DNA, and it turns out that you can find it. Not only that, you can get this high flat state, but then it can come up from this homology site because we only have 7 base pairs of homology. Then it's not quite stable enough, right? You find somebody who looks pretty good, and then a month later, it's not my soulmate. Then you come up, right? Then it turns out that you get trapped by another one next door, very close by, very similar.
Is that her sister?
Huh? And then you can oscillate. Until you find a better match with an 8, 9, 10 base pair homology and so on. So that is what we propose in that paper. And so there was a movie posted online on YouTube by students in Columbia University. So let me try to play that movie. Speed dating. So the students who made this movie told me that they spent 1 week making it. So they used tapes to stick these little items on the whiteboard in the lab. This is the whiteboard background. And there was another paper by Steve Korskowski's lab in UC Davis, published in Nature earlier that year, and they proposed yet another way to accelerate the homology search. And you have seen this in the movie. It's called intersegmental transfer. You can see that an intermediate where the Recce filament binds to 2 parts of the DNA simultaneously. So if I want to make the analogy again, so you can actually sample a different potential partner while you are still in relation with someone else. So that way you can make the process faster. Okay. All right. So another thing that we did, I worked with Paul Selvin several years ago to put together a single molecule laboratory manual published by the Cold Spring Harbor Laboratory. So this sold pretty well. So using the proceedings from the book, I was able to buy 2 dining sets in my house. So every time somebody buys this book, I get $4. And so in the first introduction of the book, I rewrote that single molecule techniques are still difficult to master, and major research findings are coming from a small number of research groups. And this will change as the methods become more commoditized. Basically, it becomes easier to use and just commercialized, and anyone can do it, and so on. So that's the direction we are also pursuing, because that way, single molecule methods can not only be used to study systems that have been studied for the last 10 years, but we can use this method to really participate at the beginning of a new enterprise. When a new gene is discovered, becomes its function, then you can really jump in and help out at the very beginning. So that's one vision. And the other is that in terms of futures, technical frontiers will be in extremes of 2 different kinds. One is ultra high precision in vitro investigation that, as you guys know, that we are pursuing, and then of course to do things in live cells and even entire organism level investigations. All right, so I stop here. I made this slide many years ago, so I don't have all the new people's names, but so these are the people who used to be in my lab and who are in my lab now, and then all of the collaborators and PIs. Thank you.